In previous lecture, we discussed about the structure, activation and deactivation of G protein coupler receptors. Another very important class of receptors is receptor tyrosine kinases. These receptor tyrosine kinases are present in the plasma membrane at the surface of the cell. Another type of tyrosine kinases, they are present within the cytoplasm, and those kinases, they play a very important role in the signal transduction. And those kinases, they are called as the cytoplasmic tyrosine kinases. But here these receptors, they are present and in the plasma membrane, and as I said, they are kinases. Kinases, as you know, are the enzymes which transfer the phosphate group from one molecule to another molecule. But specifically here in this case, these kinases, they transfer this phosphate group from the ATP and attach it to the tyrosine amino acid of the polypeptide chain. So they will pick up the phosphate group from the ATP and will attach it to the hydroxyl group of this tyrosine amino acid of the substrate polypeptide chain, and that is why they are called as tyrosine kinases. So they receive the signal from the extracellular side and transfer this signal from the plasma membrane towards the cytoplasmic side, and this transfer, this signal transduction from the plasma membrane towards the cytoplasmic side involves their kinase activity. Let us first try to understand the general, most common structural features of these receptor tyrosine kinases. So far as the structure of these receptor tyrosine kinases is concerned, it is a single polypeptide chain. Mostly it is a single polypeptide chain, but in case of insulin receptors, we have two polypeptide chains. But most common feature of these receptor tyrosine kinases is that they are single polypeptide chains. And as you know, like any other polypeptide chain, it is having two terminals. One is this C terminal and another is this N terminal. C terminal, as you know, is having the free carboxyl group, and N terminal, as you know, is having the free amino group. C terminal is towards the cytoplasmic side and N terminal is towards the extracellular side. And this orientation is decided at the time of protein translation, which occurs at the surface of this endoplasmic reticulum. When the ribosomes get translocated at the surface of endoplasmic reticulum, this orientation is decided there at the time of protein translation. So we are having this C terminal towards the cytoplasmic side and N terminal towards the extracellular side. In between these two terminals, we have two important segments. One segment is called as this alpha helix segment, and this alpha helix segment is inserted in the plasma membrane. And here in this case, in the receptor tyrosine kinases, we have only one alpha helix. As you remember, in case of the GPCRs, we were having 7 transmembrane alpha helices, but here we are having only one transmembrane alpha helices, and this alpha helix structure is composed of hydrophobic amino acids. So we are having this single alpha helix. So this receptor passes through the plasma membrane only by a single time. So this is the alpha helix structure which is inserted within the plasma membrane. Then towards the C terminal present within the cytoplasmic domain, we are having this tyrosine kinase domain. This tyrosine kinase domain shows the enzymatic activity. This is the domain which shows the enzymatic activity. It phosphorylates the tyrosine amino acids. So we are having this tyrosine kinase domain, then we are having this alpha helix domain, and these two domains are present between the C terminal and N terminal. And remember that this tyrosine kinase domain showing the enzymatic activity is its intrinsic property, like cytokine receptors, which we will discuss in coming lectures. In cytokine receptors, we are having other enzymes that get associated with this receptor in the cytoplasmic side, and there in the cytokine receptors, the enzyme activity is not the intrinsic property of those cytokine receptors, but here it is the intrinsic property. This property of it is this kinase activity is its own, it is intrinsic property. So this is the tyrosine kinase domain, and then we are having this alpha helix domain. This alpha helix is inserted within the plasma membrane. So this is the most common structural feature of these receptor tyrosine kinases. They are composed of single polypeptide chain, and in this single polypeptide chain we have this alpha helix segment which is inserted within the plasma membrane, and then we are having this tyrosine kinase domain which is present within the cytoplasm. Now the question is how this receptor is activated when a signaling molecule binds to the binding site present in the extracellular side of this receptor. It involves dimerization, and that dimerization causes the increased tyrosine kinase activity of the cytoplasmic site. First, a signaling molecule as a ligand binds to the binding site present in this extracellular side of this receptor. Here we have these two receptors very close to each other. So, signaling molecule will first bind to this receptor at this binding site, here in this case. This binding will cause a conformational changes in this receptor. Those conformational changes will increase the protein-protein interaction with this neighboring receptor, and this receptor is also the tyrosine kinase receptor. So these two receptors will come very close to each other, and this close protein-protein interaction will lead to the formation of these dimers, and this is called as the dimerization. This signaling molecule binding conformational change induced dimerization will increase the tyrosine kinase activity of this tyrosine kinase domain, and this increased activity will cause this autotronous phosphorylation. That is, tyrosine amino acid is present in this polypeptide chain in the cytoplasmic side will get phosphorylated, and this kinase domain will phosphorylate the tyrosine amino acid is present in this polypeptide chain of this receptor, and simultaneously this domain will phosphorylate the tyrosine amino acid is present on this chain. So they will crisscross this phosphorylation. This segment will phosphorylate the tyrosine amino acid is present in this chain, and this will phosphorylate the tyrosine amino acid is present in this chain. So it is called as the autotronous phosphorylation. It will not only phosphorylate their own tyrosine amino acids, but they will also phosphorylate the tyrosine amino acids present in its neighboring chain. So this is the autotronous phosphorylation. First we have this ligand binding, this signaling molecule binding. Then we have this dimerization. This dimerization is of two types, either receptor induced or it is ligand induced. In case of this receptor induced, we have a signaling molecule that binds to a single receptor at a time. These are monomers. They have single polypeptide chain. This single polypeptide chain signaling molecule, it binds to a single receptor. Once it binds to this receptor, it causes conformational changes in this receptor. And because of these conformational changes, it will increase the protein-protein interaction with this receptor and will lead to the formation of the dimer. So this is called as receptor-induced dimerization. And this kind of receptor-induced dimerization occurs in case of EGF, epidermal growth factors, because these EGFs, they are monomers, they are having a single polypeptide chain, and they will bind only one receptor at a time. And dimerization induced by this EGF is the receptor induced dimerization. Another case is when a signaling molecule is composed of two polypeptide chains and it simultaneously binds to both these receptors. That is, these signaling molecules, they are having two polypeptide chains and they simultaneously bind to these two receptors, and this dimerization is induced by the ligand by itself. That is why it is called as the ligand induced dimerization, and this dimerization occurs by PDGF or NGF, platelet derived growth factor or nerve growth factors. So these signaling molecules, these growth factors, they are having two polypeptide chains, and these signaling molecules bind, these growth factors bind simultaneously to two receptors, and this binding leads to dimerization. Once they form the dimers, because of the conformational changes, and it leads to the increased tyrosine kinase activity of these tyrosine kinase domains. Once the enzyme activity in this segment or in this segment gets increased, they will not only phosphorylate their own tyrosine amino acids, but they will phosphorylate the tyrosine amino acids of the neighboring receptor molecules, and this will be called as the transphosphorylation. So here we will be having their tyrosine amino acids that will get phosphorylated by the tyrosine kinase activity of this segment of this receptor. So this will phosphorylate the tyrosine amino acid is present in this segment, and this kinase activity will phosphorylate tyrosine amino acid is present in this segment. So we will be having auto and transphosphorylation. Here you can see the phosphorylation occurs in this catalytic site, and here in this case it is the non-catalytic site. So this catalytic site as well as non-catalytic site will be now having tyrosine amino acids which are phosphorylated. So this increased catalytic activity not only phosphorylates the tyrosine amino acids present in the catalytic site, but it also phosphorylates the tyrosine amino acids present in the non-catalytic site. This increased activity, this increased tyrosine activity which occurs because of the conformational changes induced by the signaling molecule binding and then leads to the formation of these dimers. This increased activity phosphorylates the tyrosine amino acids. These tyrosine amino acids present in the catalytic site as well as non-catalytic site are phosphorylated. Now they have phosphoryl group attached to this hydroxyl group of this tyrosine amino acids. Now the question is how the signal from this receptor, from the cytoplasmic side of this receptor gets transducted or gets transferred to the cytoplasmic side. In the cytoplasm, we have some specific target receptors, target proteins which bind to the cytoplasmic side of this receptor. And those receptors, those receptor proteins, those target proteins will bind to the cytoplasmic side of this receptor only when there are phosphotyrosine amino acids. Then those target proteins, those proteins will bind only to the phosphotyrosine amino acids. That is we have in the cytoplasm some specific proteins which have binding sites specifically for phosphotyrosine amino acids. And these proteins, these are the target proteins which have binding site for phosphotyrosine amino acids. And usually these proteins have binding site that is called as the SH2 domain, or they have PTB domains, phosphotyrosine binding domains, and SRC homology 2 domains. In the cytoplasm, we have some specific proteins. These proteins have some specific binding sites for these phosphotyrosine amino acids which are present in this non-catalytic site. From here, these proteins will transfer this signal from these receptors to the downstream proteins, downstream target proteins. But here in these proteins, they have binding sites specifically for these phosphotyrosine amino acids present in the non-catalytic side of this receptor. So the sequence is like this: have signaling molecule binding to the binding site present in the extracellular side. That leads to the dimerization of this receptor. This conformational changes induce dimerization, activates the tyrosine kinase activity of the cytoplasmic domain of this receptor. This increased catalytic activity phosphorylates the tyrosine amino acids in the catalytic site as well as in the non-catalytic site of this receptor. The phosphorylation of this tyrosine amino acid present in the non-catalytic site will now provide the binding site for these proteins. These proteins have specifically some domains like SH2 domain or PTB domains. They are specific for phosphotyrosine amino acids, and they bind to these phosphotyrosine amino acids, and once these proteins bind to this phosphotyrosine amino acids, they will then transfer their signal to the downstream target protein. This is how this receptor tyrosine kinase receptor gets activated and how it transfers the signal to the cytoplasmic side of the cell.